And we're just going to give a little bit more time for anybody else to join us. Okay, hopefully we'll see if more people attend, but we'll go ahead and start for now. Um, Hello, everyone. Thank you for joining us today on our ALS Webinar Wednesday series. My name is Alex Yao. I'm the marketing coordinator for ALS North America, and I will be facilitating the webinar today. Before we begin the presentation, I have a few helpful notes. If you're having technical issues with our webinar platform or any questions, type your message or question using the chat function at the bottom of the screen. You can also select the hand icon to notify me of your status so I can assist you. All questions regarding the webinar material will be answered at the end of the presentation. Our presenter today is Rui Sun. Rui has a Master and Ph.D. degrees in Analytical Chemistry from the University of Missouri. She has been with BioScreen Testing Services, now part of ALS, for 8 years and serves as Chemistry Manager at ALS-BioScreen in Torrance, California. Rui, thank you for your time to present for us. I'll now turn it over to you.
Well, thank you, Alex, for the introduction, and thanks to everyone for joining the webinar. Today, I will be talking about elemental impurities in pharmaceutical and personal care products. So first, I would like to everybody know that the information and views expressed in this presentation are solely from myself and do not represent any official perspectives of ALS Global Corporation, BioScreen Testing Services, or other companies or regulatory agencies. So here is the outline for my presentation. We will go into why elemental impurities is important in pharmaceutical and personal care products. And we'll also go into regulations and guidelines on elemental impurities testing in pharmaceutical and personal care products. And these regulations and guidelines are from different agencies, including ICH, US FDA, EMEA, USP, EP, Health Canada, and California Proposition 65. Then I'll talk about analytical methods for elemental impurities testing. And at the end of the presentation, I'll be taking questions. So first of all, why is elemental impurities testing important in drug products? And the main reason is that elemental impurities pose toxicological risk to patients without providing any therapeutical benefit. So their levels in the drug product should be controlled within acceptable limits. However, elemental impurities may be present in drug products, and they are from a variety of sources. The first possible source is that there are elements intentionally added in the production of drug product components. For example, platinum and palladium are frequently used as catalysts in pharmaceutical industry. And the second possible source is elements present in the ingredients used in the preparation of a drug product. And another possible source is elements that leached into drug substance from container closure systems or from manufacturing equipment. Another question is why elemental impurity testing is important in cosmetic products. So similarly to drug products, the main reason is that many elemental impurities in cosmetic products may cause injury to the health of the user. So their levels in the cosmetic products should be controlled within acceptable limits. And elemental impurities may be found in cosmetics due to the persistent nature of these elements and the fact that they're found in the natural environment. Elemental impurities are found naturally in environment in rocks, soil, and water. And since the pigments in the cosmetic products may be from these natural sources, so elemental impurities can be found in the pigments and other raw materials used in the cosmetic industry. And some of these elemental impurities have been used as cosmetic ingredients in the past. For example, thimerosal is a mercury-based preservative and has been used in many cosmetic products. And FDA has banned the use of mercury compounds in all cosmetics except those that are used around the eye areas. Another example is progressive hair dye lead acetate. And in 2018, FDA published a final rule to repeal approval for the use of lead acetate as a color additive in hair dye due to safety concerns. So in the next few slides, I would like to go into regulations and guidelines on elemental impurities testing in pharmaceutical products. So I'll be presenting information mainly from ICH, FDA, EMEA guidelines, and also from USP and EP chapters. So in recent years, FDA, ICH, USP, and industry experts developed new standards that are in alignment and help ensure high quality medicines to best protect patients from the risks posed by elemental impurities. And these guidelines provide limits for elemental impurities in drug products and standardized approaches to use to determine the elemental impurities levels in the drug products. Specifically, these guidelines provide information on a variety of subjects, including the evaluation of toxicity data from potential elemental impurities, the establishment of a permitted daily exposure for each element of toxicological concern, and application of a risk-based approach to control elemental impurities in drug product. And finally, testing strategies, analytical procedures, and the requirements for method validations. So as I briefly mentioned in the previous slides, elemental impurities may be present in drug products from a variety of sources. So now I'll go into a little more details about the potential sources of elemental impurities in drug product. So here is a diagram that shows an example of typical materials, equipment, and components used in the production of a drug product. Some potential sources, for example, drug substance and excipients, they could contain elemental impurities resulting from the elements intentionally added from the catalyst in the production of this drug substance or excipients. So the risk assessment of the drug substance should address the potential for inclusion of elemental impurities in the drug product. In some cases, elemental impurities are not intentionally added, but they're still potentially present in the drug substance, water, or excipient that is used in the preparation of the drug product. And the other potential source we need to consider is elemental impurities that can be potentially introduced into the drug substance or the drug product from the manufacturing equipment, and that can be leached from the container closure system. So each of these sources may contribute elemental impurities to the final drug product through any individual or any combination of the potential sources. So during the risk assessment, the potential contributions from each of these sources should be considered to determine the overall contribution of elemental impurities of the final drug product. Now we will get into classification of elemental impurities in drug products. So in ICH, FDA guidelines, and USP elemental impurity chapters, the elemental impurities have been placed into 3 classes based on their toxicity and likelihood of occurrence in the drug product. And class 1 includes arsenic, cadmium, mercury, and lead, which are very toxic to human, and they have very limited or no use in the manufacture of pharmaceutical products. Their presence in the drug product typically come from commonly used materials such as excipients. But because of their unique nature, evaluation of these 4 elements is required during the risk assessment across all potential sources and all routes of administration. And the outcome of the risk assessment will determine those components, and it may require additional controls. And one of the ways for controlling the elemental impurities is to include testing for these class 1 elements. And class 2 elements are generally considered route dependent, and they're also toxic to human. Class 2 elements are further divided into subclasses 2A and 2B based on their likelihood of occurrence in the drug product. Class 2A elements, they have relatively high probability of occurrence in the drug product. So risk assessment of class 2A elements is required across all potential sources of elemental impurities and routes of administration. Class 2B elements have a reduced probability of occurrence in the drug product related to their low abundance and low potential to be co-isolated with other materials. And as a result, they may be excluded from the risk assessment unless they are intentionally added during the manufacture of drug substance, excipients, or other components of the drug product. And Class 3 elements have a relatively low toxicity by oral route of administration, but may require consideration in risk assessment for inhalation and parenteral routes. So I also like to point out other elements such as aluminum, boron, calcium, iron, potassium, magnesium, manganese, sodium, tungsten, and zinc. If these elemental impurities are present or included in the drug product that are addressed by other guidelines and or regional regulations and practices that may be applicable for particular elements or quality consideration for the final drug product. So on this slide, we have a table here from ICH guidelines that provides recommendations for which elemental impurities should be included in the risk assessment. And this table can be applied to all sources of elemental impurities in the drug product. But based on their routes of administration, the company can determine which elements should be included in the risk assessment. The risk assessment can be facilitated with information about potential elemental impurities provided by the suppliers of drug substances, excipients, container closure systems, and manufacturing equipment. The data that support this risk assessment can come from a number of sources, including prior knowledge. For example, if there is prior knowledge of elemental impurities, concentration range from a specific sources, it would be very helpful to include that information in the risk assessment. And other sources can be published literature, data generated from similar processes, and supplier information or data. For example, if the elemental impurities testing results in the drug substance and excipients can be provided by the suppliers, that would be helpful for the risk assessment. And finally, testing of the components of the drug product and testing the final drug product. Now I wanted to get into some information provided by the guidelines regarding analytical procedures for elemental impurities testing. So first, I'll start with the statement in the ICH guideline regarding analytical procedures for elemental impurities testing. So ICH guideline states that the determination of elemental impurities should be conducted using appropriate procedures suitable for their intended purposes. And the test should be specific for each elemental impurity identified for controlling during the risk assessment. And pharmacopoeia procedures or suitable alternative procedures to determine the elemental impurities levels should be used. And there are two frequently used pharmacopoeia procedures, which are USP 232 and 233 elemental impurities, and EP 2.4.20 determination of elemental impurities. I will go into more details of these two procedures in the next few slides. So USP 233 elemental impurities procedures chapter describes two analytical procedures for the evaluation of elemental impurities levels. Procedure 1 is elemental impurities analysis by inductively coupled plasma atomic or optical emission spectroscopy, ICP-AES, or also called ICP-OES. And procedure 2 is analysis by inductively coupled plasma mass spectrometry, SPMS. So this USP chapter also provides acceptance criteria for alternative procedures. And basically, alternative procedures that meet the validation requirements listed in this chapter can be used for elemental impurities testing. This USP chapter also provides options for sample preparations for elemental impurities analysis, including neat direct aqueous solution, direct organic solution, indirect solution, and closed vessel digestion. So neat injection is used for liquid samples that's suitable to be introduced to ICP-OES or ICP-MS systems without any additional sample preparation steps. And if alternative procedures are used, such as atomic emission spectrometer, atomic absorption spectrometer, and X-ray fluorescence analysis, neat injection may be used if it's suitable for the sample. And direct aqueous solution is used when the sample is soluble in an aqueous solvent. So generally for ICP-OES and ICP-MS analysis, direct aqueous solution or direct dilution with nitric acid is preferred for the sample preparation due to minimal interferences. So also hydrochloric acid, hydrofluoric acid, sulfuric acid, hydrogen peroxide, or a combination of the acid are also commonly used to dissolve the sample for ICP-MS or ICP-OES analysis. And direct organic solution is used where the sample is soluble in an organic solvent. However, when using this sample preparation procedure, it's important to be careful with a couple of things. First, we would try to use the highest purity of the organic solvent if possible, since elemental impurities analysis is usually preferred, performed at a trace level. We would like to minimize any potential contaminations from the reagents. The other thing that is important to consider is the potential matrix effects and interference that may be caused by the use of organic solvents. An indirect solution is used when the material is not directly soluble in aqueous or organic solvents. So the total metal extraction is preferred when sample preparation approach to obtain the indirect solution. And finally, closed vessel digestion is a sample preparation procedure that is designed for sample that need to be digested in a concentrated acid using a closed vessel digestion systems. And closed vessel digestion minimize the loss of volatile impurities. The choice of a concentrated acid will depend on the sample matrix. So at the bottom of the slides, here are two examples of microwave digesters that are commonly used for closed vessel digestion. In the next few slides, I will be talking about USP requirement for method validation of analytical procedures for elemental impurities testing. So USP 233, elemental impurities procedure chapter states that by means of validation studies, analysts will confirm that the analytical procedures described in this chapter are suitable for use on specified material. If the specified compendial procedures, ICP-OES and NMS, do not meet the needs of a specific application, an alternative procedure may be developed. Alternative procedures must be validated and shown to be acceptable by meeting the validation requirements for the alternative procedures. So the requirements are different for validation of limit procedures and quantitative procedures. So for method validation of limit procedures, the validation parameters should include detectability, precision, specificity. And for the validation of the quantitative procedures, the validation parameters should include accuracy, precision, specificity, limit of quantitation, range, and linearity. And in the precision section, we should include both repeatability and intermediate precision. So for method validation of limit procedures, to demonstrate detectability, we'll need to prepare and analyze a standard solution at 100% target concentration, a spike sample solution 1 at 100% target concentration, a spike sample solution 2 at 80% target concentration, and unspiked sample solution. And to meet the acceptance criteria for non-instrumental procedures, the spike sample solution 1 must provide a signal or intensity equivalent or greater than that of standard solution. And spike sample solution 2 must provide a signal or intensity less than that of spike sample solution 1. And to meet the acceptance criteria for instrumental procedures, the average value of 3 replicate measurements of the spike sample solution 1 must be within plus minus 15% of the average value obtained for the replicate measurement of the standard solution. And the average measurement of the replicates of spike sample solution 2 must provide a signal intensity or value less than that of the standard solution. For method validation of limit procedures to demonstrate precision, we will need to prepare 6 independent samples spiked with appropriate reference materials for target elements at target concentration. And the acceptance criteria is that the relative standard deviation must be not more than 20% for each target element. And for non-instrumental procedures, specificity, the validation study, and for non-instrumental procedures, the precision is demonstrated by meeting the detectability requirement. And to demonstrate specificity, the validation study must show the procedure is able to unequivocally assess each target element in the presence of components that may be expected to be present, including other target elements and matrix components. So this can be done by spiking the sample with appropriate levels of other target impurities and matrix components and demonstrate that these impurities are determined with appropriate accuracy and precision. And this also can be done by confirmation by other supporting analytical procedures. So for method validation of quantitative procedures to demonstrate accuracy, we would need to prepare solutions containing target element at concentration ranging from 50 to 150 of the target concentration, and prepare samples spiked with appropriate reference materials at concentrations ranging also from 50 to 150% of the target concentration for each target element. And the acceptance criteria is that the spike recovery must be within 70 to 150% for the mean of 3 replicate preparations at each concentration. So for method validation of quantitative procedures to demonstrate precision or repeatability, we would prepare 6 independent samples taken from the 6 lot, spiked with appropriate reference materials for the target concentration, which is usually at 100% of the target concentration. The acceptance criteria is the relative standard deviation must be not more than 20% for each target element. To demonstrate intermediate precision, the repeatability analysis will need to be performed again either on a different day with a different instrumentation with a different analyst or a combination of those, and then combine the results of this analysis with the repeatability analysis. So the total number of is 12. The acceptance criteria is that the relative standard deviation must be not more than 25% for each target element. So for the method validation of the quantitative procedure, the specificity requirement is the same as for the validation study of limit procedure. A limit of quantitation range and linearity is demonstrated by meeting the accuracy requirement. So now I'm going to get into EP chapter a determination of elemental impurities. So in general, there are a lot of similarities between USP and EP chapters for elemental impurities determinations. However, we will see some differences between these two compendial chapters in terms of the approach for sample preparation, measurement procedures, and the requirement for method validation. So first of all, EP chapter states that as a reference procedure is not provided for each element, matrix, and concentration. The choice of the procedure is the responsibility of the user. However, flowcharts are provided in the EP chapter to determine sample preparation and measurement method. So I'll be presenting the flowcharts in the next couple of slides. And EP chapter also states that all suitable sample preparation methods and measurement techniques, such as atomic emission spectrometry, atomic absorption spectrometry, X-ray fluorescence spectrometry, ICP OES, and ICP-MS, can be used for the determination of elemental impurities if the method has been verified before use by a system suitability test or a validation procedure according to the EP chapter. So now we're going to go into sample preparation, measurement, and validation requirement in this EP chapter. So instead of providing specific options for sample preparations like USP chapter does, EP chapter gives this nice flowchart to help us determine sample preparations procedures. So first, we would ask the question, is the procedure described in the monograph for the material to be tested? And if the answer is yes, then we will need to ask the second question. Is the sample suitable for direct analysis? And if the answer is yes, then we would go directly to measurement. And if the answer is no, we will need to prepare sample solutions. And depending on the outcome, we could proceed to measurement, or we'll have to go back and verify the procedure. And on the left side of this flowchart, if the answer to the very first question is no, there's no procedure described in the monograph for the material to be tested, then we will need to ask further questions about the sample to determine if the sample is suitable for direct analysis or need to be prepared in aqueous solution, organic solution, or if it needed to be digested in an open or closed vessel system. Once the sample preparation procedure is determined, we can then proceed to the next flowchart to determine how to measure the elemental impurities in the sample. So the first question we need to ask here is that if there is a measurement method described in the monograph for this material to be tested. And if the answer is yes, then we can proceed to perform analysis using the monograph procedure. Depending on the outcome of the recovery of the standard solution, we can report the result, or we have to go back and verify the measurement system. And similar to the decision tree for the sample preparation on the left side of this flowchart, if the answer to the very first question is no, there is no measurement method described in the monograph for this material to be tested, we will need to develop a test procedure and validate this procedure and make sure the validation requirements are met before we can use the procedure for testing the sample. And then, of course, when we test the sample using the validated procedure, we'll need to make sure that the system suitability requirements are met before reporting the results. Now let's get into EP validation requirements for analytical procedures of elemental impurity testing. So here is the general statement in the EP chapter regarding a method validation. Before the initial use of the selected procedure, the analyst must ensure the sample preparation and measurement method are appropriate for the elements of interest, sample matrix, and instrument used. This is accomplished by following the validation procedure before the initial use and the system suitability test on the day of analysis. So similar to USP, the requirements are different for method validation of limit procedures and quantitative procedures. So for method validation of limit procedures, validation parameters should include specificity and limit of detection. And for the method validation of quantitative procedures, the validation parameters should include specificity, range, accuracy, repeatability, intermediate position, and limit of quantitation. So for method validation of limit procedures, to demonstrate specificity, EP requires the validation study must show the ability to ensure that the analytical procedure allows a reliable determination of the elements of interest in the presence of components such as carrier gas, impurities, and matrix that may be expected to be present. So this is demonstrated by complying with the accuracy requirement for elements to be determined. And to determine the limit of detection, we will need to determine the lowest concentration, giving a signal clearly distinct from that obtained with a blank solution. And the acceptance criteria is that the limit of detection is not more than half of the concentration of the specification limit. For method validation of quantitative procedure, the requirement for specificity is the same as for the limit procedures. And the range is demonstrated by complying with accuracy requirement for elements to be determined. For method validation of quantitative procedures to demonstrate accuracy, we would spike the sample with a known concentration of a reference standard of element of interest at 3 concentration levels in the range of 50 to 150% of the specification limit, and each concentration is performed in triplicate. And the acceptance criteria is that spike recovery must be within 70 to 150% for the average at each concentration level. And to demonstrate repeatability, we'll spike 6 independent samples with suitable reference standard at 1 concentration level, or 3 concentration levels at each concentration level in triplicate. And its acceptance criteria is the relative standard deviation is in both cases must be not more than 20%. So for method validation of quantitative procedures to demonstrate intermediate position, we would perform repeatability analysis on different days or with different instrumentation or by different analysts. And please note that only one of the 3 experiments is required. And the acceptance criteria is that the percent RSC must be not more than 25%. And the EP approach for limit of quantitation is to use the results from the accuracy study and determine the lowest concentration meeting the accuracy criteria. And the acceptance criteria for limit of quantitation is that the limit of quantitation is below the specification limit. So far, we have been focused on the regulations and guidelines in elemental impurities testing in pharmaceutical products. And now I would like to talk about some regulations and guidelines on heavy metal impurities in cosmetic products. So currently, there are no international standards for impurities in cosmetic products. Limits have been established in Germany. Rather than taking a risk-based approach like the pharmaceutical industry, the German limits are based on the levels that could be technically avoided. So elemental impurities were limited to anything above the normal background levels. And the German government conducted tests to determine background levels of heavy metal contents in toothpaste and other cosmetic products. And based on their studies, it was determined that heavy metal levels in cosmetic products above the value listed below are considered technically avoidable. So for lead, it's at 20 ppm, arsenic at 5 ppm, cadmium at 5 ppm, mercury at 1 ppm, and antimony at 10 ppm. Now let's get into Health Canada guidance on elemental impurities in cosmetics. So many heavy metals are not acceptable as ingredients in cosmetic products sold in Canada, as they may cause injury to the health of the user. So Health Canada has taken a similar approach to the German limits in the establishment of heavy metal impurity limits, as the department has always maintained that impurities in the cosmetics should be reduced to the extent that is technically feasible. So the metals of primary toxicological concern in cosmetics are lead, arsenic, cadmium, mercury, and antimony. And Health Canada heavy metal impurity limits in the cosmetic products are for lead, not more than 10 ppm, for arsenic, not more than 3 ppm, cadmium, not more than 3 ppm, mercury, not more than 1 ppm, and antimony, not more than 5 ppm. So other metals such as selenium, barium, and chromium may be present as impurities in the cosmetic products. However, the toxicological properties and corresponding risks associated with this substance are considered less significant than for lead, arsenic, cadmium, mercury, and antimony. And accordingly, the impurity limits in the cosmetics have not been developed for these metals. So now let's take a look at US FDA's testing of cosmetics for elemental impurities. So FDA has surveyed a variety of cosmetics on the market, testing for arsenic, cadmium, cobalt, lead, mercury, and nickel. And the results are published on the FDA website. And now I put a link to the website here if you're interested in getting more information on this topic. So a few questions we might ask here. Why did FDA conduct surveys of the cosmetics on the market? So this type of surveys help FDA find out whether there are problems that require FDA's action to protect the public health. So what metals are selected for these surveys? The metals that were selected are arsenic, cadmium, chromium, cobalt, lead, mercury, and nickel. So these are elements that are present naturally in the earth, and they're among a group of substances commonly referred as heavy metals. How did FDA conduct these surveys? So the strategy was to cover a wide variety of cosmetic products and a broad price range. And FDA contracted with outside labs to do the analysis for the survey. And what did they learn from the surveys? So first, the amount of arsenic, cadmium, chromium, cobalt, lead, mercury, and nickel that they found were, for the most part, very small. And they currently do not have information indicating that the amount they found would pose a health risk, which is a good news for the consumers. And also the surveys showed that the products such as eyeshadows, blushes, and compact powders contain more heavy metals than other type of cosmetic products, which suggests that most of these elemental impurities in the cosmetics come from minerals used as pigments and as fillers. So FDA. So now let's look at what are the FDA regulations and guidance that address the elemental impurities in cosmetic products. Similar to the limits in cosmetic product limits in Germany and Canada, these limits are not established on a risk-based approach. So for mercury, it is only allowed in cosmetic as a preservatives in the eye area products. And the mercury must be not more than 65 ppm in the finished product. And only if there's no other effective and safe preservative is available. And mercury is not allowed in any other type of cosmetic product except in a trace amount that is less than 1 ppm, and only if its presence is unavoidable under good GMP. For lead, FDA has published a draft guidance for the industry that recommends a maximum level of 10 ppm as impurity in cosmetics. And this guidance applies to cosmetic lip products such as lipsticks, lip glosses, and lip liners, and externally applied cosmetic products such as eyeshadows, blushes, shampoos, and body lotions marketed in the United States. For arsenic, lead, and mercury in color additives, the limits are arsenic not more than 3 ppm, lead not more than 20 ppm, mercury not more than 1 ppm. And chromium hydroxide green and chromium oxide greens are both permitted for the use as color additives in externally applied cosmetics, including use in the eye area. And the listing regulation for the color additive limits of chromium as an impurity is not more than 50 ppm. So since I'm located in California, I would also like to briefly talk about California Proposition 65. Proposition 65, officially known as Safe Drinking Water and Toxic Enforcement Act of 1986, was enacted as a ballot initiative in November 1986. So when you're in California, you probably have seen some form of the warning signs like this in a store, on a label of something you purchase, or in a resort. That's because Proposition 65 requires businesses to provide warnings to Californians about significant exposures to chemicals that cause cancer, birth defects, or other productive harm. So these chemicals can be found in the products that you purchase, or in your home or workplace, or that are released to the environment. The Proposition 65 list contains a wide range of naturally occurring and synthetic chemicals that are known to cause cancer or birth defect or other productive harm. And this list, which is updated at least once a year, has grown to include about 900 chemicals since it was first published in 1987. So I would like to point out here is that some elemental impurities are on the Proposition 65 list, which include arsenic, cadmium, lead, mercury, and nickel due to their toxicity. So in the next couple of slides, I would like to give a brief review of analytical methods for elemental impurities testing. For non-instrumental methods, there was a USP Chapter 231 heavy metals, which is a visual comparison procedure. And the content of the metallic impurities in the sample are colored by sulfide ion and the specific test conditions. And lead nitrate is used as a reference standard that's also colored by sulfide ion. And by visual comparison of the color of the solutions, it will determine if the heavy metals in the sample exceed the limits. So I'd like to point out the substance that typically will respond to this test are lead, mercury, bismuth, arsenic, antimony, tin, cadmium, silver, copper, and molybdenum. So this test is widely acknowledged to be inadequate in terms of scope, accuracy, sensitivity, and specificity. So the chapter has been replaced by USP 232 and 233 elemental impurities, which were discussed earlier in this presentation. EP also has a heavy metal chapter that provides a similar visual comparison procedure. This chapter still can be used for the determination of elemental impurities if the method has been verified before use by system suitability test or validation procedure. And more commonly, instrumental methods are used for elemental impurities testing because of the better accuracy, sensitivity, and specificity. So now I will give a brief review of some of the instrumental methods. First, inductively coupled plasma atomic or optical emission copy, SCP-AES or SCP-OES. So the sample solution is pumped into the system where it's nebulized, forming a fine sample aerosol. Then the aerosol is carried into a high temperature argon plasma and is atomized in the plasma. So the AES uses a quantitative measurement of the optical emission from the excited atoms to determine the analyte concentration. So on the right-hand side, here's an example of one of the SCP-OES instrument models that we have here available at ALS-BioScreen. So another very commonly used instrument for elemental impurities testing is inductively coupled plasma mass spectrometry, SCP-MS. So very similarly to SCP-OES, the sample solution is pumped into the system and where it's nebulized, forming a fine sample aerosol, and the aerosol is then carried into a high temperature argon plasma and is atomized and ionized. And the mass spectrometer uses quantitative measurement of the mass to charge ratio to determine the analyte concentration. And here is a picture of one of the SCP-MS instrument models we have here at ALS-BioScreen, which has been used for a lot of elemental impurity testing projects for pharmaceutical and cosmetic products. Another instrument that can be used for elemental impurity testing is atomic emission spectrometry. It's a technique to determine the concentration of an element in a sample by measuring the intensity of one of the emission lines of the atomic vapor of the element generated from the sample. And the determination is carried out at the wavelengths corresponding to this emission line. And similarly, elemental impurity testing can be done by atomic absorption spectrometry, which is a technique to determine the concentration of an element in a sample by measuring the absorbance of electromagnetic radiation by the atomic vapor of the element generated from the sample. And the determination is carried out at the wavelength of one of the absorbent lines of the element of interest. So the picture here is the AA instrument model we have here at ALS-BioScreen. And X-ray fluorescence analysis is another option for elemental impurities analysis. XRF is based on the measurement of the X-rays emitted by the atoms of the sample when it is excited by an external source of radiation. XRF is suitable for liquid, solid, and powdered materials and is widely used as a means of screening pharmaceutical ingredients and products for elemental impurities, for quality control and in-process testing. It is also used to identify inorganic foreign elements within falsified pharmaceutical products. So to summarize my talk today, elemental impurities testing is important for pharmaceutical products because elemental impurities pose toxicological risks to patients without providing any therapeutic benefit. So their levels in the drug product should be controlled within acceptable limits. And elemental impurities may be found in cosmetics due to the persistent nature of these elements and the fact that they're found in the natural environment. And many elemental impurities in the cosmetic products may cause injury to the health of the user. So their levels in the cosmetic product should be controlled within acceptable limits. ICH, FDA, USP, and EMEA guidelines present processes to assess and control elemental impurities in the drug product. Both USP and EP chapters describe general approach and analytical procedures for the determination of elemental impurity levels. And the method validation requirements are also described in those compendial chapters. FDA and Health Canada regulations and guidance address some heavy metals as cosmetic ingredients or impurities in cosmetic products. And there are a lot of analytical procedures that can be used for elemental impurities testing. So that concludes my presentation today, and thank you very much, everyone. Please feel free to ask any questions. And I also want to share some selected references here. I'd like to say that a lot of the informations in this presentation are from these references. And also, I would like to thank my colleagues here at ALS-BioScreen, Reniel Fernando, General Manager, Wenyi Chang, Operations Director, Mitch Herbal, Technical Director, and Sharita Johnson, Chemistry Manager. And here is my contact information. So again, thank you very much, everyone. And I'll turn this back over to Alex now.
Thanks, Ray. I'm going to open up the forum for questions now. So please write your question using the Q&A function in your toolbar at the bottom of this webinar. Attendees can also see all submitted questions using that window. And if you're comfortable speaking, please indicate when submitting your question, and I can call on you so you can voice your question directly to Ray. I'll give you guys a few minutes to type in your questions. And while you guys are doing that, I'll go over some brief notes. This webinar presentation has been recorded. All participants of this webinar will receive a follow-up email once the recording is available to view. We'll also post our webinars on our ALS Global website and ALS YouTube channel in the webinar playlist. I encourage you to find a wide range of video resources and past webinar Wednesday presentations available there. Also, please follow our company page on LinkedIn as we post announcements and registration links to future webinars and other valuable resources and updates. All right, so I'm going to wait and see if we have any questions being submitted. I'll give you guys a few more minutes. All right, well, I don't see any questions. I guess Ray's covered everything. I just want to thank everybody for attending today. And if you want, you can contact Ray for any other questions later. Her contact details are right there, and they'll also be included in the follow-up email. I hope everybody enjoys the rest of their week. Thanks. Mm-hmm.